Hi everyone. Good afternoon. I'd like to welcome you all to the first seminar for this year's Organismal Biology Seminar Series. It's my great pleasure to be able to introduce Franz Goller, welcome him to McGill in Montreal. So I've known about Franz's work for many years now. We've used his models of thinking about the physiological processes that are important for the generation of complex communication signals in songbirds for many years, and it's been very informative to have all the experiments, the data from the experiments that he's generated. So Franz has had a very diverse and prolific background. He got his PhD from the University of Notre Dame, where he worked with Harold Esch, looking at thermoregulation in bees. And then he did his postdoc with Rod Suthers at Indiana University. That's where he started working on songbirds, looking in particular on the biomechanics of song production. And since then, he's been continuing that line of work, and his work focuses primarily on how the nervous system interacts with peripheral structures like the respiratory system and vocal musculature to produce these complex vocalizations. And for his studies, he integrates across many different levels of analysis. He integrates physiology with both computational biology as well as behavioral ecology. And he's using these sort of approaches to study evolutionary variation in vocal production. Now, he's published with many different authors, and most recently he's published with his daughter on behavioral thermoregulation in lizards, which is pretty neat. But without further ado, I'd like to welcome Franz.
Thank you very much, Jon, for first inviting me and hosting me. It's been wonderful to be here and talk to so many interesting people today, so thank you for putting up with me and your hospitality. I have 3 more kids. My dream is that we'll have a paper, all 5 on the author list, but I don't know how to integrate the computer gaming man into this. At any rate, so avian vocal diversity, or the acoustic signals birds make, are clearly very diverse, and I am not playing you a symphony, I'm just showing you a few representative pictures of them. And so I don't think I have to convince people of that. What some of you may or may not know is that the avian vocal organ, which is different from the mammalian organ, it's not the larynx, it's the syrinx and sits on the other end of the trachea, the windpipe. The avian vocal organ has been studied since probably the mid-18th century, and quite extensively in the 19th century. And people saw that there's a lot of morphological diversity in the avian vocal organ, and they used it as a taxonomic characteristic to classify birds. And they've speculated on sound production mechanisms, but that was speculation. They didn't have the tools to really study the functional morphology of these different vocal organs. And I'm trying to tell you sort of a few stories how we are attempting to fill in this gap in our knowledge. Birds, like humans, when they sing, they use multiple motor systems, and respiration being, of course, one. They drive an airstream past the vocal organ, the syrinx, which sits in this respiratory system at the basal end of the trachea, where it splits into the primary bronchi. And if you've watched the bird sing, you've also seen that it moves the beak and the throat seems to puff, so there's upper vocal tract filtering. Just like in humans, except the vocal organ is different. Most of the research on birdsong has been done in Oscine songbirds, and the reason for this is that Oscine songbirds learn song in a process that is reminiscent of how human infants learn speech. And to make this possible, we have brain circuitry that is dedicated to this vocal behavior, especially 4 brain areas that are dedicated to vocal learning and production. And people have figured out a motor pathway, which is these 4 brain areas, and the acronyms are not important for my talk, but there's sensory motor cortex-like areas that are dedicated to song production in songbirds. And then there's a second pathway, the anterior forebrain pathway, and these areas are required for song plasticity. That is, song plasticity during the development process, and if we have adult plasticity. So a lot of focus was on this central control of song, and because only vocal learners have this forebrain circuitry, many other groups of birds have not been studied to the same extent. The vocal organ, and this is again a schematic of a songbird vocal organ, Austrian songbird vocal organ. It's modified cartilage structures that are covered up by muscles that control other cartilages and play a role in controlling acoustic features of the sound. And the respiratory system of birds is different in that we have air sacs as bellows and a fairly rigid lung, unlike the mammalian lung. And the syrinx sits in one of these air sacs. So unlike the mammalian larynx, the syrinx actually sits in airspace. So what I would like to do today is talk about one acoustic parameter that we think we understand the best at the moment, and that is how do birds control sound frequency, and sort of take you through a few smaller stories that illustrate that morphology is important, so the morphological characteristics of the sound-producing structures will be important. But in Austrian songbirds, we always talk about neural control, and so we'll see how these two interact in a few stories. And then I'm going to switch gears and tell you, put you to sleep, basically. I'll tell you a song dreaming story very quickly at the end. Okay, so what do we sort of know about control of sound frequency? It's complex. This is it. Okay, so fundamental frequency is illustrated here. It depends on the tension of the vibrating structures, and all these parameters affected, and I will be talking about the sound source, of course, and the histological composition of the vibrating structures is going to be important, how it reacts to strain, and how stress then gives rise to tension and varying sound frequencies. Syringeal movement stands here for neural control aspects, because central neural control, of course, affects these movements. And some of them are passive, induced by neural control, that is respiratory. So the pressure that the respiratory system generates affects how the vibrating structures are situated in the vocal organ and thus affect the tension. And we'll talk about these. I will not talk about feedback because that's at the moment all theoretical. We haven't found a good experimental approach to testing these theories. Okay, so this is the syrinx again of an Austrian songbird. We take a frontal section through it, and then you see something like this. And what I've illustrated here is in blue. The darkness of the blue indicates pressure levels. So during expiration, pressure, subsyringeal pressure is higher, right, driving air out of the system. But because the vocal organ sits in an air sac, the actual vibrating structures in Austrian songbirds, we call them labia, they are surrounded by higher pressure, whereas through the vocal organ you have the pressure differential that's necessary to drive self-sustained oscillations. So pressure is pushing these labia into the airstream from the outside as well as through the pressure differential across the syrinx. On inhalation, of course, the pressure situation is reversed, and I will not be talking about inspiratory vocalizations, although some birds can do that. In terms of neural control, we'll look at how these muscles contribute to sound frequency. And when I was a postdoc, we did a study on the brown thrasher, and we looked at EMG activity of this muscle, the ventral syringeal muscle, and we found this beautiful relationship, very tight for a physiologist, between fundamental frequency and EMG activation. So we thought this is the main muscle that tenses the labia and thus controls frequency. And I was talking to somebody today about thinking we had it all figured out, and I thought that's it. But then we looked at the zebrafish, very different relationship, totally unexplained high frequency sounds in terms of muscle activation. That's a long story I won't be able to go into. But basically, neural control of frequency means that muscles tense the labial structures in whatever relationship there may be. Okay, so first story, morphological specialization. My impression is that Austrian songbirds have very little morphological variation of the vocal organ if you just look at it, right? What meets the naked eye is not very diverse, but so these are the Aussies, within the order Passeriformes. Then there are other groups. The Acanthicitidae are the New Zealand wrens, which are the most basic group of Passeriformes, and they are near extinction, so I haven't got my hands on them yet, although I would like to. But then we have all these groups that we call sub-Aussines, which are mostly American, but mostly South American, Central American in terms of their distribution. Thousands of species, but the one North American group that is represented here are the tyrannid flycatchers. They belong to the sub-Aussines. You look at their syrinx morphology and wow, they all look different. Okay? So we'll look a little bit into this. And here are some representative pictures and families within the sub-Aussines. The Tyrannidae are here. So we have the ovenbirds, wood creepers, ant wrens, ant birds, and ant wrens, etc. Okay, so mostly Central and South American. And in the old literature, what you see is that people noticed these differences, of course, and they said, wow, these groups are tracheophones because the sound source is obvious, as I will show you in a second, and it's obviously located in the trachea, whereas these groups are bronchophones because the sound source seems to be located where the trachea has split into the primary bronch. And if we look at these tracheophones, this is what you immediately see. These, we call them membranous windows or just windows, are very striking features of all tracheophone syringes. They vary in terms of how much of remnant cartilage or ossified cartilage they contain. So these cartilage rings of the trachea end here, and then this is just the membrane in this Synclotus species. The cartilage, main cartilages of the trachea also end here in the ovenbird, but there are little remnants of cartilage in this membrane. They are no longer attached to the main cartilage on each side. Okay? And just to remind you, this is what an Aussine syrinx looks like, right? Very different, no such window here in the trachea. So people thought this window is the sound source in these tracheophones, and this is illustrated in a cartoon here. So here you have the trachea, the tracheal cartilages, here are the two windows, and this is the bronchial septum. So this is the pessulus, as it is called in technical language, where the two bronchi start in midline in the syrinx. Okay? And the idea was these membranes are set into vibration by the passing airstream, and this is the sound source of the tracheophones. We wanted to test this because I had looked in my Aussine-centric view at this syrinx once in Argentina, and thought that I saw more sound sources down here like Austrian songbirds have, and we wanted to test this. And the way we tested this is by inserting a fiber optic, fine fiber optic. We illuminate the area, and then we cannulate an air sac of the bird, and inject air into it, and we can induce artificial phonation, and then we can see what vibrates. It's as simple as that. Even in a high-tech world like ours, one look may do the trick. So if you look down, I want you to orient you a little bit. If you look down like this, this is what you see. Okay, so what we see are the windows here, these windows, you see here and here. And then you look down onto this bronchial septum, which is this line here. And the dark areas are the bronchi, you're looking down into them. And what you see down here are these white structures. That's what I recognize as a possible sound source from looking at the osseous surround. So we want to test this. So we insert this fibroscope and then induce phonation. And just to give you still pictures, here the two windows are coming together. So are these labia down here. And here the two membrane windows are together. And I'm going to show you this in a movie so you get an idea of what we're looking at here. So initially the fibroscope is above the window, and then I want to test whether the window is the sole sound source, and I push through the window area and look at the labia. And then we ask, is there sound coming out? Okay, the windows are coming together, yes? Sounding awful. And now we push through and just look at the labial area. We still get sound. It sounds different. A beautiful sound source here, the labia. And then we pull the fiber back a little bit, just to, now the window again. Okay? So, we propose that these birds actually have three sound sources. The membranous window in the trachea, and two labial sound sources, one in each bronchus, as indicated. One is indicated in green here. And it's interesting that they sort of are perpendicular to each other when they meet and vibrate. I don't know whether that has any significance. I just found it cute. So to test this, we have used ovenbirds. This was the distress call, and I hold them in my hand. And this is the normal one, when the bird is fully intact. What you see is the blue line, or can't see because it's not very rich in contrast, is amplitude modulation of a particular frequency, fairly loud call, lots of harmonic contents. Some low frequency contribution here in the spectrum, and when we then disable the ventral membrane by either poking a hole in it, or as I will show to you in a second, by putting tissue glue on it and making it rigid, then we get a change in the acoustic structure, something like this. We lose a lot of the low frequency, it sounds different. You can't hear the amplitude modulations anymore, for example. And so we repeated this with the rupture of the membrane. So the first one was glue, this is rupture, and we lose these high frequency elements. So our proposal is that the three sound sources probably work simultaneously in these birds, most of the time, at least for this call, and that the membrane vibrations are low frequency and modulate the amplitude of the labially generated sounds. Okay, so that's the proposal. So in summary of this first story, tracheophones actually have three sound sources, we think. Now, how independently they can work, we don't know yet. Okay, so that's probably going to require a lot more work. And these ovenbirds are very sensitive. They don't do well in captivity. They share something of the psychology of the Argentine friends. It's the state bird of Argentina, so very aptly chosen. At any rate, we think the membrane windows do the low frequency portions of the sound, and the left and right labia, the higher frequency. We don't know whether the two labial sources can work independently. That's all hopefully something for the future. They definitely interact in complex ways, giving rise to these amplitude modulations and complex spectra. What we also know from a few other species that we've begun to study, one ant bird, for instance, in Panama, seems to rely on the membrane windows more than the oven bird in Argentina. And we have an ant shrike from Argentina where we did this same thing. And it uses it the way the ovenbird does, so the labial sound source is modulated by the window. So this variation in how many cartilages are in this window and whether they're still attached to the framework or not probably gives a morphological basis to different acoustic signatures of these different birds. Okay, story 2. Let's look at neural control of sound frequency. And in Austin's, we knew for a long time from people who denervated the syringeal muscles that the fundamental frequency drops of their songs. They don't make high frequency sounds, suggesting that the muscles are responsible for tensing labia. We didn't know anything about sub-Austin's. And here we studied terality, and especially the kiskadee at first. And the kiskadee sounds like its name says, kiskadee, kiskadee. And here we have subsyringeal air sac pressure measured along with the sound. The horizontal line is ambient pressure, everything below is inspiration, everything above is expiration. And the Tyranid syrinx doesn't have the membrane window either, right? It's a different family of subaustin. It has prominent ventral muscles on the syrinx. We denervate these muscles by performing nerve cuts on either side of the trachea here, so there's no neural activation of these muscles, and the kiskadee sounds the same. All frequencies are present. Basically indistinguishable dependence on air sac pressure, which suggests that kiskadees modulate the frequency of their song by altering the pressure that drives it, that you sort of see in these gray bars you would have if I had pointed it out, that there's a beautiful correspondence between pressure and frequency, and across all three syllables. Okay? So it doesn't seem like they use their muscles to do so. Whereas an Austin, the Austin, the right mouse of Austin research, the zebrafinch, not exactly known for high frequency sounds, but there are a few in here if you look. After the nerve cut, they're all gone. These high frequency elements are now all harmonic stacks. Okay? So here we definitely see neural control, and if we look at the pressure fundamental frequency relationship, this is what we find in intact birds. So muscular control contributes a lot to making high frequency. When we denervate the zebrafinch syrinx, we find this relationship. This is how much pressure drives frequency in the zebrafinch, and it is about 70 hertz per kilopascal. So not that much, it's a small change. Okay? Now in kiskadees we didn't have calibrated pressure, so I can't tell you whether it's a different relationship or just the same, and we are working on that. Okay, if we talk about neural control, I should point out that syringeal muscles are among the fastest vertebrate muscles known. The other super fast muscles that we know all have to do with sound production, toadfish, swim bladder muscle, but here the muscle makes the sound pulse, right? Each contraction is a sound pulse. And rattlesnake tail shaker muscles have been described as the super fast muscles within vertebrates. These syringeal muscles, here you see a histological prep with the fast oxidative fibers stained in dark red. All the other fibers don't stain for any myosin node. Okay, we term them super fast fibers. So we use sort of negative evidence to show how prominent they are in these syringeal muscles. And these syringeal muscles can modulate sound at 260 hertz in starlings, up to 200 hertz in zebrafinches. Very, very rapid. Try that. Okay, 200 times a second. Good luck. Okay, so this was the kiskadee and the zebrafinch. We started, a graduate student of mine, Sarah Garcia, started doing a big comparative project, and what I'm showing you is her data on the fox sparrow and the western kingbird, the tyrannid, and an Aucene, of course. And when you do nerve cuts, here's the frequency histogram for intact fox sparrow song and calls. Everything the bird said is included in this histogram. You do a right nerve cut, you drop out a lot of high frequency components. You do a left one, you drop out a lot of high frequency components. You do a bilateral cut, all the high frequencies are gone. Okay? What I'm showing you here is this is not just true for the learned vocalizations like song. We think calls in these birds that have functions like alarm or contact or distress, typically not learned, but they also affect it. So they use neural control for their calls too, whereas the western kingbird, this is 2 syllables out of its song here, no change after any nerve cut, just like the kiskadee. Okay, and we've got more and more species added to this. So far, all tyrannidae we've tested have not been using their muscles to control sound frequency. Okay, so a big difference here, and the next step is we've got the syringes extracted from all these sub-Aucenes that we studied in Argentina. We haven't been able to export them yet, but the next step will be that we actually look at morphological differences to see how much they contribute to different frequencies within the tyrannid. Okay, so I made a big case that neural control is big in Aucenes, pressure controls frequency in tyrannids. We have morphological difference and different sound sources in other sub-Aucenes like the Philnali-Eadie. And now the next question I would like to address is, to what degree does the makeup of the vocal organ affect the frequency an Aucene can sing? So neural control is fine, it still has to use the vibrating structures, so to what degree does that affect it? And I'm beginning to tell this story with the starling, where we measured airflow through the primary bronchi. From these flow signals you can tell which side contributes which frequency to the song, and so the sound is illustrated here as a spectrogram. Flow through the right side, flow through the left side, and the air side pressure that you've already seen. So all the sounds are made during expiration. The inspirations are shaded in gray here. And let's look at this first sound. We look at the flow, no flow through the right side, flow through the left side. We conclude this is made on the left side. And by this, we can go through the large vocal repertoire of the starling and figure out, for instance, that this high-frequency sound has flow to the right and almost no flow to the left, so we conclude this is made on the right. And with this, we can sort of determine the frequency range of each side. Yes? Okay, now let's look at morphology of the labia. So here we have the frontal section through the syrinx that we've already seen, and this blue box shows the details of the medial labia, so the medial partner of each pair, right? On the left, on the right, and on the left. So the right medial labium and the left medial labium. Right? You get the point already. It's so clear. It's 8 times bigger on the left than the right, so that it can cover lower frequency ranges than the right is pretty obvious. So there's a clear morphological correlate to this range difference. But interestingly, starlings resort to a trick to make very low frequencies. Starlings can make 250 hertz to 800 hertz sounds. They're all very rich in harmonic content, and they make them, we think, with pulse tone, a pulse tone mechanism, which is a different mode of vibration that has been described in people as vocal fry. And often it's seen as a pathology. And at that point, my vocal folds are closed for a very long time, open briefly and close, and slap into each other. And when these is far from a sinusoidal vibration, so we have a lot of rich energy content in upper harmonics. and the Australians do the same thing. But I don't have more time to go. So we wanted to do a comparative study across Austin species to look at, do we see overall across species a correlation between the makeup of the labia and the frequency ranges these birds can cover through neural control. And so we did the typical stains for collagen, elastin, and hyaluronic acid. The labia are made up like the vocal folds in people out of extracellular matrix with these elastic proteins. There's no muscle in the core like in the human vocal fold in birds, but other than that, it's pretty much a convergently evolved structure. And so we do this, and then we look at the orientation of the fibers, and we come up with cartoon images of the arrangement of these elastic proteins. And what we find is that in some species, it's very homogeneous. All the fibers seem to be oriented throughout the whole depth of the labium the same way, like in the zebrafinch. And then in other species, it seems like there are layers, where collagen fibers either in higher proportion in some part than another, but also oriented in different orientations in some part than another. And this has also been described in human vocal folds, and the idea is that perhaps we as humans can recruit different portions of this vocal fold into vibration, and through this layered structure. And so if that is the case, then if we have layered structure here, perhaps we have the same thing happening. Just another, in our comparison, we wanted to do closely related species. For the birders among you, the golden crown and the ruby crown kinglet, both about 6 to 7 grams, both very small. Same genus, different species, and very different songs. That's the golden crown kinglet, very high pitched. And the frequency of its song is depicted here on the histogram. And the ruby crowned likes to show off with a deep voice. Much lower the song, but also now they're migrating, so if you go out, they're probably some in the trees, and you can spot them by this call. And you wouldn't attribute this call to a 6.5 gram bird.